Hello everyone, my name's Iman. Welcome back to my YouTube channel. Today we're going to tackle a practice problem set that relates to our lecture on non-enzymatic protein function and protein analysis. Let's go ahead and get started with problem number 1. Problem number 1 says, at what pH can protein A best be obtained through electrophoresis? Note that MM means molar mass. So here in this table we have 3 proteins, A, B, and C. We're given their PIs and their molar masses. Now remember, electrophoresis separates proteins based on charge. So for a protein to move through the gel, it needs to be charged. If the pH of the gel is equal to the protein's PI, then the protein is going to have no net charge and it will not move. That means we can't separate it from the rest. So keeping that in mind, to isolate protein A using electrophoresis, we want it to be charged while ideally minimizing the movement of the other proteins. One way to do that is to adjust the pH so that protein A is charged and it migrates, while protein B and C do not move. Protein A, it has a PI of 4.5. So at a pH above this, it's going to migrate towards the positive electrode. Looking at the answer choices, option D is pH 5.5. This is slightly above protein A's PI, which gives it just enough negative charge to move through the gel. Meanwhile, at pH 5.5, protein B is near its PI at 6, so it'll be nearly neutral and it won't migrate much. And then protein C, it has a PI of 9.5. So it's going to be strongly positively charged at this pH. So it's going to move in the opposite direction. So at a pH 5.5, protein A will migrate the most, while the others are going to remain mostly stationary and they won't interfere. So that makes D the best answer for problem number 1. Let's go ahead and move into problem 2. Problem 2 says, what is the function of SDS in SDS-PAGE? Let's read through the answer choices. A says SDS stabilizes the gel matrix, improving resolution during electrophoresis. B says that SDS solubilizes the protein to give them uniformly negative charges so that the separation is based purely on size. C says SDS raises the pH of the gel, separating multi-unit proteins into individual subunits. And D says SDS solubilizes proteins to give them uniformly positive charges, so separation is based purely on pH. Now remember, SDS-PAGE is a type of gel electrophoresis that's used to separate proteins based purely on size. But that only works if we first eliminate differences in charge and shape. That's where SDS comes in. SDS is a detergent that binds to proteins and then denatures them. That means it unfolds the protein and it removes any effects of shape or folding. But more importantly, SDS coats the protein with a uniform negative charge. So no matter what the protein's original charge was, after SDS treatment, every protein behaves like a long, negatively charged rod. That makes the separation dependent only on size, not charge, not shape, just how big the protein is. So with that information, it becomes obvious that the correct answer for problem 2 is B. Let's move on to problem 3. Problem 3 says, which of the following is not involved in cell migration? Let's break this down by looking at what each of these choices does. Choice A says dynein. This is a motor protein that moves along microtubules. It's involved in the sliding motion of structures like cilia and flagella, which help with cellular movement. So yes, it plays a role in cell migration. B says flagella. Flagella are tail-like structures that help propel cells forward, especially in sperm cells. So that's definitely a direct form of cell migration. C says actin. Actin forms microfilaments, which are essential for cell crawling and shape changes during migration. So actin is very much involved in cell movement. Finally, D says centrioles. These are different. They're involved in cell division, not cell migration. They help organize microtubules during mitosis, and they form the spindle apparatus, but they don't directly contribute to cell movement. So the correct answer here is going to be D, because this is the only one that's not involved in cell migration. Problem 4 says, which of the following proteins is most likely to be found extracellularly? Let's go ahead and work through the answer choices here as well. A says tubulin. Tubulin is the building block of microtubules, which are part of the cytoskeleton. This is found inside the cell, and it plays a role in cell structure, transport, and division. It's not outside the cell, so this is not what we're looking for. B says myosin. This is a motor protein that interacts with actin in muscle cells, and it's intracellular transport. Again, this is an intracellular protein, not what we're looking for. C says collagen. This is actually our correct answer. Collagen is a structural protein that's found in the extracellular matrix. It provides strength and support to connective tissues like skin and cartilage. So collagen is not just found outside the cell, it is one of the main proteins in the extracellular space, which is why this is the correct answer for this problem. Let's still work through D. D says actin. Actin forms microfilaments, and it's critical for cell movement, shape, and intracellular transport, but it's located inside the cell, which is not what we're looking for. So once again, the only protein in this list that is extracellular is collagen. Problem 5 says, hormones are found in the body in very low concentrations, but tend to have a strong effect. What type of receptor are hormones most likely to act on? Statement 1 says ligand-gated ion channels. Statement 2 says enzyme-linked receptors. And statement 3 says G protein-coupled receptors. This problem is really testing our understanding of how hormones work. Hormones don't just flood the body. They're released in small amounts, but they can still cause big effects. That means their receptors need to be highly sensitive and able to trigger amplification of the signal. Let's think about ligand-gated ions first. These open or close when a ligand binds, and that allows ions to flow in or out of the cell. They respond quickly, but they don't generate a large intracellular signaling cascade. So while they are important in things like neural signaling, they're not typically used by hormones. What about enzyme-linked receptors? These are actually excellent hormone receptors. When the hormone binds, it activates an enzymatic domain inside the cell, which often leads to a cascade of phosphorylation events. And this leads to signal amplification, which means a small signal causes a big cellular response. Finally, there's G protein-coupled receptors. These are also common targets for hormones. A single hormone binding a GPCR can cause a strong and widespread effect from a small stimulus. So 2 and 3 are true statements, which means that the correct answer for problem 5 is C. Problem 6 says, which of the following is most likely to be found bound to a protein in the body? To answer this, we need to think about which ions are commonly tightly regulated or stored in bound form, especially by carrier proteins or buffering systems. So let's go through the options. A says sodium. Sodium is a monovalent cation, and it's mostly free in solution. It's tightly regulated by ion channels and pumps, but it's not commonly found bound to proteins in a stable way. B says potassium. Well, just like sodium, potassium is a freely dissolved monovalent ion in the cytosol. It plays a major role in membrane potential, but it isn't typically carried by proteins. C says chloride. Chloride is an anion that balances positive charges in cells and tissues. It's regulated by channels, not by binding proteins. Finally, we have calcium. This is the correct answer. Calcium is a divalent cation, and its concentration in cells is kept extremely low in the free form. To prevent toxic buildup and to control calcium-dependent processes, The body uses calcium binding proteins like calmodulin and others. And these proteins hold calcium in a bound form until it's needed for signaling or contraction. So the correct answer for problem 6 is going to be D. Problem 7 says, which of the following characteristics is not attributed to antibodies? So here we're looking for the one false statement among the 4. Let's go through each option. A says antibodies bind to more than one distinct antigen. That's false and our correct answer. Each antibody is highly specific and it binds to only one unique antigen. That's what gives antibodies their targeting precision. So they don't bind multiple different antigens. They're designed for one target only. Let's still work through the other answer choices. B says antibodies label antigens for targeting by other immune cells. This is true. Antibodies can act like flags. They bind to pathogens and then signal other immune cells to come destroy the target. C says antibodies can cause agglutination by interaction with antigen. This is also true. Because antibodies have 2 binding sites, they can bind to multiple identical antigens and cause clumping or agglutination, which helps the immune system clear out pathogens more efficiently. Finally, D says antibodies have 2 heavy chains and 2 light chains. That's correct. Antibodies are made of 4 polypeptide chains, 2 identical heavy chains and 2 identical light chains. So the only incorrect statement here is A, making that the correct answer. Let's move on to problem 8. Problem 8 says, which ion channels are responsible for maintaining the resting membrane potential? The resting membrane potential is the baseline electrical charge across a cell's membrane, and it's usually around 70 millivolts in neurons. This potential is mostly established by the constant movement of potassium ions out of the cell. So which channel is responsible? The answer is ungated channels. Ungated channels, especially potassium leak channels, are always open. So they allow potassium to move out of the cell down its concentration gradient. This movement of positively charged ions out of the cell makes the inside more negative, and it helps maintain the resting membrane potential. So these channels are passive and they're continuously active, which is exactly what's needed for a stable resting state. So the correct answer here is A. Problem 9 says, which of the following is not a component of all trimeric G proteins? The term trimeric means 3 parts. All trimeric G proteins are made of 3 subunits, alpha, beta, and gamma, and these 3 work together and they're found in all trimeric G proteins. That means that the correct answer is D. GI is not a subunit. Instead, it refers to a type of G alpha subunit. So this is not required across all trimeric G proteins, hence why it's the correct answer for problem 9. Now we move on to problem 10, which says, which of the following methods would be the best to separate large quantities of the following proteins? The table we're given shows 3 proteins, A, B, and C. They all have very similar isoelectric points, but their molar masses are quite different. So when we're choosing a method here, we want to pay attention to 2 key clues that are given to us in this question stem. The goal is to separate large quantities, and the biggest difference among the proteins is in their molecular weight, not their pi. So let's look at the answer choices. A says ion exchange chromatography. This separates proteins based on charge, which depends on their isoelectric points. But here, the pi values are too close to be helpful, so this is not the correct answer. B says size exclusion chromatography. This is actually the best answer. It separates proteins based on size, allowing larger proteins to elute first and smaller ones to elute later. And then since the molecular weights are significantly different, this method will actually effectively separate them. But let's still work through C and D. C says isoelectric focusing. This technique also separates based on PI, and since the PI values here are so close together, it won't be effective here. There's only a 0.3 difference between B and C, and there's only a 0.2 difference between A and B. So this is not going to be as efficient. D says native page. Now, while this does separate based on size and charge, it's better suited for analytical purposes and not for large scale separation. Also, it's less precise for preparative purification. So the best method to separate large amounts of proteins that differ mainly in molecular weight is going to be size exclusion chromatography. So 10 is B. Let's look at problem 11. Problem 11 asks, which amino acid contribute most significantly to the PI of a protein? To answer this, we need to remember that the isoelectric point, or PI, is the pH at which a molecule carries no net electric charge. For proteins, the PI is heavily influenced by the ionizable side chains of the amino acids that make up the protein. So let's go through the three choices we're given. The first is lysine. Lysine has a basic side chain with a positively charged amine group. So it contributes significantly to a protein's overall charge and therefore affects the PI. Then we have glycine. Glycine is neutral. It has no ionizable side chains. It just has a hydrogen atom as its R group. So it contributes very little to the PI beyond the backbone amine and carboxyl groups, which all amino acids have. Finally, there's arginine. Arginine also has a basic positively charged side chain. And so like glycine, it strongly influences the protein's PI. So 1 and 3 are the correct choices, which makes the correct answer for 11 C. Problem 12 says, how does the gel for isoelectric focusing differ from the gel for traditional electrophoresis? A says isoelectric focusing uses a gel with much larger pore sizes to allow for complete migration. B says it uses a gel with SDS added to encourage a uniform negative charge. C says isoelectric focusing uses a gel with a pH gradient that encourages a variable charge. And D says the gel is uncharged in isoelectric focusing. The protein is treated before loading. Now, let's remember the core differences between these two methods. Traditional electrophoresis, like SDS-PAGE, separates proteins based on size. Proteins are treated with SDS, which gives them a uniform negative charge. Then they move through a gel towards the positive electrode, and their migration is based on molecular weight. But isoelectric focusing is different. It separates proteins based on PI, or the isoelectric point. This is the pH at which the protein has no net charge. And to do this, the gel must contain a pH gradient. So when an electric field is applied, proteins migrate through the gel until they reach the region where the pH equals their PI. And at that point, they stop moving because they carry no net charge. So the key feature of the gel in isoelectric focusing is the presence of a pH gradient. And answer choice C captures that exactly. So 12 is C. Problem 13 says, which protein properties allow UV spectroscopy to be used as a method of determining concentration? A says proteins have partially planar characteristics in peptide bonds. B says globular proteins cause scattering of light. C says proteins contain aromatic groups in certain amino acids. And D says all organic macromolecules can be assessed with UV spectroscopy. Now, the answer here for problem 13 is C. Proteins contain aromatic groups in certain amino acids. So when using UV spectroscopy to measure protein concentrations, we rely on the fact that certain amino acids absorb UV light, specifically the aromatic amino acids like tryptophan and tyrosine, and to a lesser extent, phenylalanine. These absorb strongly at 280 nanometers, which is why absorbance at this wavelength is commonly used to estimate protein concentration. And this property is unique to these aromatic side chains, not just to the fact that the molecule is organic or that it has peptide bonds. So the only option that explains why UV spectroscopy works for this purpose is answer choice C. Let's move on to problem 14. Problem 14 says a protein collected through affinity chromatography displays no activity even though it is found to have a high concentration using the Bradford protein assay. What best explains these findings? A says the Bradford reagent was prepared incorrectly. B says the active site is occupied by free ligand. C says the protein is bound to the column, and D says the protein does not catalyze the reaction of interest. Now, the Bradford assay measures total protein concentration based on the binding of the dye to amino acid residues, but it doesn't assess whether the protein is active. That means the protein could be present, but not functional for several reasons. One likely explanation is that during affinity chromatography, the protein may have co-eluted with excess free ligand used in the purification process. If this ligand is still bound to the active site of the protein, then it would block substrate binding, which would effectively render the enzyme or protein inactive, even though it's present in solution. So the best answer for problem 14 is going to be B. Now we can tackle our last and final problem. Problem 15 asks what property of protein digesting enzymes allows for a sequence to be determined without fully degrading the protein. So the key concept here is partial digestion. You want to break the protein into predictable fragments and not completely destroy it. This ability to target specific cleavage sites is called selectivity. It means the enzyme will only cut at certain amino acids, and it will produce fragments that help reveal the original sequence without chopping up the entire protein into unusable pieces. And that's why the correct answer is A. And with that, we've completed this practice problem set. I hope it helps. Please let me know if you have any questions, comments, concerns down below. Other than that, good luck, happy studying, and have a beautiful, beautiful day, future doctors.